Welcome back to the Sand Science Experience, where we explore the most exciting new research papers and preprints just for you. Before we dive into today's topic, please take a moment to like this video and subscribe to our channel. It really helps support the Sand Science Experience. Okay, so today we're exploring something really unexpected. It's about how a major mitochondrial deficiency, specifically complex 1, affects the liver. What's surprising here is we're looking at Leigh syndrome, which, most people think of as primarily hitting the brain.
Right. And what's truly remarkable about this research is, how it puts the spotlight on the liver's role. It's often underestimated, in the whole picture of this systemic disease. It kind of challenges some long-held ideas about where the real problems lie in disorders like Leigh syndrome.
Absolutely. So our mission for this deep dive is basically to unpack the liver's surprising metabolic resilience, but also reveal these kind of hidden disruptions happening under the surface in a mouse model missing complex 1, and crucially, connect that to what it means for the whole system. Ready? Okay, let's start with the core finding because it's pretty striking. In this NDUFS4 knockout mouse model, it's the main model for Leigh syndrome they used here, the liver had a huge drop in complex 1 activity, like 86% reduction. You'd instinctively think that's catastrophic for energy, right?
You certainly would, yeah. But the fascinating thing they found was, despite this severe hit to complex 1, the liver's overall cellular respiration, it stayed largely unaffected. And the reason seems to be that the liver just intrinsically relies much more heavily on complex 2 for respiration. It contributes something like 85% of the total electron flow there.
Wow. So it's got this built-in preference, almost a default pathway.
Exactly. It's like it already had a backup generator running, ready to take the main load.
So the liver just kind of shrugged off this major hit.
Yeah.
That's quite the survivor tissue. It definitely makes you rethink the standard mitochondrial playbook, doesn't it?
Yeah.
How adaptable different organs can be. But okay, here's where you said it gets really interesting. Respiration seems fine, but the study showed widespread disruptions elsewhere in its internal chemistry.
Exactly. That's the twist. While energy output seems stable, the broader metabolism was significantly altered. We saw, for instance, shifts in how the liver handles glucose. Glucose itself was down, but levels of related sugars like mannose and these UDP derivatives of glucose, they were actually elevated.
UDP-glucose, UDP-N-acetylglucosamine, those specific ones.
That's right. And this pattern suggests a shift, a move towards anabolic pathways, like building glycogen stores or fueling the hexosamine biosynthesis pathway. And that pathway connects to something called O-GlcNAcylation, which is a really important way cells use sugars to tag and regulate proteins.
Okay, so the liver isn't just passively surviving, it's actively reorganizing its internal fuel lines and signaling. What else changed in this metabolic shakeup?
Well, beyond glucose, there was a definite overload of amino acids, lots of different kinds, including aromatic ones like tyrosine and tryptophan, but also key players in one-carbon metabolism, serine, threonine, methionine, and even glutathione, which signals stress.
That sounds like the cell is under significant strain, right? Redirecting resources.
Precisely. It points to cellular stress and maybe increased demands. And then looking at the core energy cycle, the TCA cycle, and NAD plus regeneration seemed okay. Its precursor nicotinamide was even up. But other TCA intermediates were altered, suggesting, again, a significant rewiring of how the cell is managing fuel. Things like fumarate, malate, aspartate were elevated.
This sounds incredibly complex, a real balancing act inside the liver. So if we step back and connect these liver-specific changes to the bigger picture, what's the implication for the whole body, especially in Leigh syndrome?
Well, take that amino acid buildup. That accumulation, for example, can activate a key signaling pathway called mTORC1/S6K. And mTORC1 is a master regulator of growth and metabolism. Its activation has been linked elsewhere to problems like hypertriglyceridemia, high triglycerides in the blood, which has been seen in other complex 1 deficient mice.
I see. And that really hammers home the importance of the liver-brain axis, doesn't it?
Yeah.
Because you mentioned therapies targeting the liver, specifically like messing with S6K1 or restricting iron, have shown neurological benefits in these mice.
Exactly. It shows just how interconnected things are. What happens in the liver doesn't stay in the liver, especially when it comes to the brain.
So treating one organ can definitely impact another.
Precisely. And this study really flags O-GlcNAcylation and that MTO signaling pathway as super important areas for future research in Leigh syndrome. They're critical integrators linking metabolism, cell signaling, gene activity. Digging into them could give us really valuable mechanistic insights and potentially new treatment angles.
Okay, so let's sum up our deep dive. We saw the liver's Pretty amazing reliance on complex 2, letting it sidestep a huge complex 1 problem for basic respiration. But beneath that, profound metabolic changes are happening. It's this complex mix of stress adaptation and dysfunction, and it has real systemic consequences.
Which leaves us with a pretty important question, I think. How much more do we really need to map out these intricate metabolic conversations between organs, especially the liver-brain axis, to truly understand and hopefully effectively treat complex conditions like Leigh syndrome?
A very profound question indeed, something for us all to think about. We really hope you enjoyed this deep dive here at the Sound Science Experience. Please don't forget to like this video and subscribe if you haven't already. It genuinely helps us out. Thanks for joining us, and we'll see you next time.